Hi everybody. Today we are going to talk on the topic denaturation and the renaturation of DNA. As the title implies itself, we are doing some experiments or we are making some studies in which something will cause the DNA to change from its original form to some other form. That is, you are changing the nature of the DNA by the action of some agents. And in the process of renaturation, you are making the DNA to come back from its altered form to its original form. So let's see how this can be achieved. Now, if you have a native double helix, this is the original form of the DNA. So here, when we apply particular heat onto the DNA, as shown in the picture, you can see that the strands start unwinding. That is, the DNA double helix will start unwinding because of the breaking of the hydrogen bonds between the two helices. And if you apply little more heat, what happens? You can see that not only the unwinding, but the DNA strands get separated. Okay. And hereafter, this process is referred to as the process of denaturation. And here we can see that if you start cooling the DNA, okay, it starts that is below a particular temperature. If you cool it, what happens? The DNA comes back to its original form, that is into the double helix form. So this change from its double helical structure to a single stranded structure is referred to as the denaturation. And the agent which we have applied here, as shown in the figure, is the heat. And the reforming of the separated strand into the double helix form is referred to as the renaturation. And the reverse of the denaturation is being used here. That is, instead of heat, we are using cooling. Now, there are different denaturing agents which can be used. Sometimes you can use temperature. So that is a temperature of 90 degrees centigrade and above can be used as the denaturing agent. Other than that, you have chemicals like NaOH. NaOH is the most commonly available and most frequently used chemical agent used for denaturing. Then you have urea, you have formamide, and also we use certain salts. Then pH, both acidic as well as alkaline pHs can also be used. So let's go in detail how each and every factor is causing the denaturing of DNA. Now coming to the heat-based denaturation, okay, here as we showed earlier itself, this process is similar to that of melting, and the DNA is first getting unwound itself, and then it becomes into separate single strands. And once the strands are separated, the DNA will then, when we cool back, we cool it back to a stable temperature, what happens? The DNA strands, the separated DNA strands will start aligning with each other. Okay, so when these process of cooling before the complete cooling has happened, what happens is, and when they are in the single stranded form, you can do some analysis also. That is, various markers or various sequences can be analyzed during this process at which the DNA is being separated as single strand. And the degeneration has been used also to compare different species. Now, for example, if two different organisms have their double-stranded DNA separated at a particular temperature, we can make an assumption that they have a similarity between each other. And what is the main reason behind this is basically this temperature at which the DNA becomes double-stranded DNA becomes separated is mainly dependent upon the G plus C content of the DNA. If the G plus C content is being found to be similar, they require a similar temperature for their denaturation and to becoming separated to from double-stranded to the single-stranded. So you can make an assumption whether the organisms or the organisms, especially based upon their DNA separation, denaturation analysis, whether they are similar or not. And this denaturation principle denaturation principle is also being used in various PCR analysis techniques also. Now this is a melting curve of DNA which has been shown. Now how can we analyze whether the DNA is being separated or not, or whether it has become a single strand? Usually DNA has an absorption maximum at 260 nanometer. So, by analyzing the concentration of a DNA in solution at 260 nanometer using a spectrophotometer, you can say the absorbance of, you can check the concentration of the DNA. And the thing that you have to note is that when the DNA becomes single-stranded, the absorbance is being found to be increasing. Okay, so you carry out a reaction in which DNA is being placed at different temperatures. Now, for example, 40, 60, 80 and 100 as shown in this graph. And then you, at each time point, that is at each temperature, you will check the absorbance. You take aliquots and check the absorbance at 260 nanometer. And that has been plotted. Okay. And at a particular temperature, now for example here in this figure, 80 degrees centigrade, you can see that the DNA, even beyond after the 60 degrees centigrade itself, you can see that the absorbance value is being found to be increasing. And then almost before reaching 100, about 90 or something, you can see that it is entering into a plateau like stage, that is, it's becoming constant. So the at temperatures beyond that, all the DNA strands have been considered to be as single strand. And at temperatures below 60, all the DNA strands will be found to be as double stranded. And hence, what happens, the absorbance will be found to be less. And when the heat is being increased, the double stranded DNA will start becoming single stranded. And simultaneously, their absorbance at 260 nanometer will also increase. And as such absorbance increases, it has been associated with the concentration of single-stranded molecules, DNA molecules, which have been formed during the process. And we use a temperature, a term called the melting temperature here. And this is the separation of this double helix, usually is called melting. And it occurs abruptly at a characteristic temperature called the denaturation temperature or the melting temperature. And it is defined as the temperature at which 50% of the DNA is being melted. You can see on the z-axis of that is on the, now in the z-axis on the right hand side of this graph, you can see the degree of denaturation is also being shown. In 50% of denaturation, at happening that at that point, what happens? You have extrapolated it and put it down, and you get a temperature of about 80. So Tm is being considered to be as here 80. So the temperature at which 50% of the original DNA becomes single-stranded is referred to as a melting temperature. And higher the Tm of a DNA, it indicates that higher the GC content of that particular DNA. Now, chemical-based denaturation. Here, we are using NaOH as an example. Other chemicals, formamide, also can be used. But the thing is that they denature DNA. They are not able to denature DNA as rapidly and or as reliably as NaOH. And moreover, NaOH can be used at a variety of concentration, and it is also can be scaled up quite easily. So because of that particular reason, we use NaOH. And moreover, once after denaturing a DNA using NaOH, you can also renature it using a phosphate buffer. Now, the DNA that is denatured through other chemicals such as DMSO, they are not able to be fully renatured in this fashion, and hence NaOH is a good candidate for chemical-based denaturation. Now, salt-based denaturation. Salt-based denaturation is something which once it is denatured using a salt, you cannot renature it. Now, this is salt-based denaturation is also being used accompanied with other heat-based methods also. Okay, now and after that, usually it is alongside of that you will also use certain other chemicals such as isopropanol and ethanol and all. And this salt based denaturing is normally done in reactions where you need to scale up and produce large amount of DNA. That is after breaking the cell and you want to produce a large amount of you want to isolate a large amount of DNA from a cell. In such cases all you will use salt based denaturing techniques. Now, another technique is a pH based method. So, at high pHs, what happens? The hydroxyl ions will pull the hydrogen ions from the base pairing, and forming a hydrogen bond between the strands, causing them to separate. Okay, so the hydrogen bonds, see, at a higher pH, okay, at a higher pH means there is more amount of OH ions. So these OH ions will have a great affinity for the hydrogen bonds and they will pull the hydrogen bonds towards them. And as a result, what happens? The hydrogen bonding between the two strands will break and the double-stranded DNA will become into single-stranded. And there are some features of denaturation as we told earlier. There is an increase of absorption of UV light, that is at 260 nanometers the absorbance of UV light, and that has been found to be increased as the denaturation of DNA happens. And this process is known as, this increase in absorbance is known as hyperchromosity or hypochromism, and this is mainly due to the unstacking of the base pairs. Then at the same time, you also have a decrease in the specific optic rotation of the DNA as well as a decrease in the viscosity. So normally to check the amount of denaturation, we use the absorption of the DNA at 260 nanometer because it can be easily conducted even in a lab. So I think this is clear to you. Now come. Thank you.